right. Do we have any questions before we get started on the new lecture? Lecture 2. No questions. All right. So this is also going to be a review lecture. So lecture 1 and 2 were both reviews. So after lecture 2, when it's done, we are actually going to get into hematology and start talking about, hardcore hematology. But again, it's very important for me that everybody starts in the same level. So we're going to go and cover these basic things. If you already know the material, sit back, listen. I'm sure you're going to learn something new during the process. And if you don't know, just ask questions and we're going to go forward. But after I'm done with this one, I just don't want to have anybody the wrong impression that this is going to be the type of things we're going to talk about in this class. The first 2 lectures is to get you started. So if you are not feeling comfortable with the material, it would be your responsibility to catch up, okay? To catch up. Because from next time, we are going to go ahead and talk about hematology, okay? All right. So we're going to talk about cell structure. We are going to talk about the basic unit of life, which is going to be cell, which means whenever you break down a cell, you're not going to have a living thing anymore, okay? A cell is going to be different types, forms, characteristics. We're going to talk a little bit about that. But in general, in most cells, especially a type of cell, we call it eukaryotic cell, is going to have certain characteristics that's going to be common regardless whatever the cell is going to be. One is that you are going to have a cell membrane. It's going to be very important. In my opinion, when life was first formed, which we have the evidence about 4 billion years ago, it was the membrane that was conducive to formation of life. Because of the fact why? There is an example here that we are having right now. If I have ability to control the thermostat, I would be able to change the temperature in this room. I don't. That's why this place is so hot. But if you come and ask me, can I go ahead and change the temperature in LA County? I cannot. You can see that. I cannot do that, but I can control the temperature in this room. Because this room is going to be encompassed within a limited space and volume, and I would be able to control it. That membrane for the cells allows the cell to control the things that are happening inside of the cell. And that's going to be the key for it. In eukaryotes, you don't have this in prokaryotes, but you do have in eukaryotes, you got a nucleus. In eukaryotes, the nucleus is going to be where you're going to have all the genetic, not all, a majority of the genetic material that's going to allow the cell to control its metabolism. When you are looking at this cell, there's nobody sitting inside of the nucleus and pushing buttons. The cell is able to control its own metabolism based on the genetic information that's available there, okay? And of course, between the nucleus and the cell membrane, you're going to have this space we call cytoplasm. A lot of things are going to be in the cytoplasm. A lot of things are going to happen in the cytoplasm. We're going to talk about those in a little bit greater detail in a few minutes. So, the cytology is going to be the study of the cells. And there was a time, that was a really good major. You guys could, especially for CLS students, they could get their, they pass the examination, they take 1 extra year courses and get their degree in cytology. And they would pay well. However, with more automation and things like that, I've heard that cytology and histology, they are more becoming automated and more being done by the pathologists. They're not going to go ahead and, they're not hiring as many as they used to be. But it was a good field, cytology. This is a pseudocoloring of a cell. You can see the 3 components that I just told you. You would be able to see the nucleus. With the nuclear membrane, you're going to be able to see the cell membrane, and you're going to be able to see the cytoplasm. If you ever seen an egg, you haven't seen a cell. That yolk is going to be the nucleus. That the white part, well, is transparent and colorless, but when you cook it, it becomes white. That protein part is going to be the cytoplasm. And in the case of the egg, the shell is going to act as the membrane. So it's very similar. It's actually the same thing, but it's larger scale. Now again, we have different types of, and I don't know why this doesn't fall on this screen, but we have different types of cells. In general, cells are divided into two general categories: prokaryotes and eukaryotes. The first evidence that we have about life goes back 4 billion years. It was prokaryotes. For about 2 billion years, and some of you don't comprehend the concept of billion. You got to think about the numbers. You got to think about, number of the zeros in front of it. For 2 billion years, life was only prokaryotes on Earth. Okay? Everything was happening at that time. Majority of the genes that are going to be in your body right now, they formed that first 2 billion years in the prokaryotes. And then they start to change as the eukaryote came into picture, and they start, you have all different varieties of these genes to be able to do different functions. Things became more complex. When we look at life, we see first life was single cell, very simple cell. If I recall correctly, the first evidence of these very simple organisms, they had about 40 to 50,000 DNA nucleotides. Do you know how many we have in each cell? How many nucleotides do we have? When I say nucleotides, I'm talking about A's, T's, G's, and C's. Do you know how many humans have it in each cell? You have about 3.5 billion of them, okay? 3.5 billion. The first prokaryotes had about 40 to 50,000. Very simple, very simple. They were from the, if you have had microbiology, mycoplasm is very similar to those original prokaryotes that came into existence. Eukaryotes are going to be slightly more complex. That's why their size is going to be bigger. So in here, the yellow is going to be the prokaryotes, while the blue is going to depict a eukaryotic cell. It's more complex. It's able to do much more different things. One of the things that happens is the presence of the nucleus. Before eukaryotes, prokaryotes did have DNA. Every living organism has DNA, but their DNA was dispersed inside of the cytoplasm. There was not a unique place like nucleus that you would be able to find the DNA in the case of the eukaryotes. In the case of the eukaryotes, that's DNA, the genetic material, was encompassed inside this membrane we call the nucleus. And we will a little bit talk about this nucleus, how it's going to be able to control the functions of the body today. Okay? But that's the story about the different cells. But a cell, and when I'm talking about the cell right now in this class, I'm just going to place my emphasis on eukaryotes. Okay? If you're going to be in microbiology course, we're probably going to keep talking to you about the prokaryotes. But hematology, we are going to be dealing with the eukaryotes, so that's what I'm going to go ahead and focus on. It's just like a city. I see a great amount of similarity between a cell and a city. You are going to have an area, as we called it, the nucleus. This is going to be the mayor's office. This is the person that's going to go and decide where the roads are going to be, who's going to be able to travel, how much is going to go and cost to travel on this road. That information is going to come from the nucleus, from the DNA. Okay? That's what is being passed on from your parents to you and from you to your children. You are going to go ahead and have sanitation workers. Sanitation workers. What's going to happen to your body? What's going to happen to your life if tomorrow everybody who is in charge of sanitation goes on strike? Maybe you survive for a day or two, but after a couple of months, you're going to have cholera, you're going to have all sorts of different diseases, and you're going to be, the diseases are going to become rampant, and they are going to be dispersed all over the population. So you have to have somebody to clean up the mess. You do have the similar structure inside of the cell. We call them lysosomes. Lysosomes are able to break down a lot of things that we want to break down. For example, you have these cells in your blood, we call them neutrophils. Neutrophils are phagocytic cells. They eat up bacteria, they eat up things that you don't need it in your body. They find them and they eat them up. They phagocyte them. What do they do with that then? What's going to happen to that bacteria that was eaten up by a neutrophil? Well, these lysosomes are going to come and fuse with this bacteria, and they're going to break them down. Break them down to what? Well, what is every living thing is made of? What is the composition of every living thing? Water? No. You can go ahead and I can assure you that if this wood is going to have water in it, but that wood is not going to be living. Carbon. This wood has carbon in it, but it's not living. Protein is one of them. Okay? And there are 4 things that living things are going to have. Every living thing is going to have. Proteins, carbohydrates, lipids, and nucleic acid. These are the 4 things that makes up living organisms. Okay? Yes, they do have carbon, they do have sodium. Those are not living things, but if they don't have the other 4, they're not going to be a living thing. Okay? So what's going to happen here is that these lysosomes are going to get those bacteria. Bacteria are living things. They're going to break them down to what? To the amino acids, to the fatty acids, to the glycerol, to glucose, to nucleic acid, thymine, adenine, guanine, and cytosine. And that's what's going to happen. They're going to recycle those bacteria. Okay? That's what's going to happen. And you're going to use the amino acid to make your own protein. So you get the guy and you destroy it, you break it down into its components, and you use the components in order to use for the metabolism of your cell. That's what happens. You would not be able to do that without lysosomes. We have diseases in which the lysosomes are going to have, there's going to be genetic mutation. These lysosomes are not going to be able to produce those enzymes that are going to be inside these lysosomes. What's going to happen? People are going to develop devastating diseases. Devastating diseases such as what? Like Tay-Sachs syndrome. Tay-Sachs syndrome is going to be a disease that's going to be genetic disease, okay? It's going to be recessive autosomal disease, which means that both parents have to contribute a defective gene to the child. What's going to happen here is that the child by the age of 2 years old is going to be deaf, blind, not be able to move, and probably by the age of 3 or 4 will die. Just because one enzyme that's supposed to work in lysosomes is not, because of the genetic mutation. As simple as that. That's Tay-Sachs syndrome, or Tay-Sachs disease in a nutshell. Mitochondria. One of the things that was different between the prokaryotes and eukaryotes is going to be mitochondria. Once upon a time, we have great amount of evidence that once upon a time, mitochondria that today we see inside of these eukaryotes was a protobacteria. It was a prokaryote itself. Some hungry eukaryotes decide to eat this prokaryote, but I call it, the way that I describe it is that it's going to give it indigestion. The big prokaryote, it's the small eukaryote, but it cannot digest it. What happens that small eukaryote has the capacity of making a lot of energy. Before that, one glucose molecule was broken down and you make 2 ATP molecules out of it. When this new guy came inside this big guy and the big guy couldn't digest it, something interesting happened. That small tiny guy inside of the big guy was able to break down the glucose and make 36 ATP. 2 ATP, 36 ATP. Which one is going to give more advantage? That's the origin of mitochondria. Once upon a time, there was protobacteria, and we exactly know how do we know that? How can I come and stand here and tell you that? This is the symbiotic relationship that developed about 2 billion years ago. Mitochondria has its own DNA. And we are able to sequence that DNA. And we are able to sequence the DNA of the bacteria that's going to be the culprit. And we're going to see the homology, similarity in the DNA so closely that there is absolutely no doubt that they are related. Okay? What does mitochondria do? Mitochondria, first of all, I'm going to talk about these different structures today. I tell you a couple functions they're going to be able to perform. But I am not claiming that I'm telling you everything about these organelles. Because something I did write half my dissertation for my doctoral degree was on mitochondria. Okay? What happens to it? What is the function of it? So I don't want you to think mitochondria makes ATP and that's the end of the story. It does a lot of things. It does a lot of things, from the calcium homeostasis to be able to maintain the calcium the right amount, to control the cell death. Sometimes cells are going to die one way or another way is all going to be controlled by mitochondria. But overall, when you're looking at the general function of mitochondria, is production of energy. Okay? Production of energy. Then, if you put things in such things in perspective, interesting concept is going to rise. Let me give you one of these concepts right now. Does anybody know why most of the mutation, genetic diseases that's going to affect the mitochondria, why does it affect the central nervous system? Why does it affect your brain and your spinal cord and your nerves that are going to come from it? Why they are affected most of any other place in your body? Does anybody know why mitochondrial diseases are going to have such a drastic effect on the nervous system? When you drown in water, somebody comes and takes you out after 10 minutes. They pronounce you dead. Is your heart dead? No. Are your muscles dead? No. Is your skin cell dead? No. What part of your body is dead? Your brain is dead. Why? Lack of oxygen. Well, why doesn't my muscle die whenever I'm trying to run a marathon and I'm in bad shape, and the day after a marathon I cannot walk because it hurt all over my body? What happened? Because I didn't have enough oxygen, I kicked into the anaerobic respiration and I produced lactic acid. So why doesn't my brain do that? Brain cannot. Brain cannot use any source of energy except for glucose. And when the glucose is going to be broken down, it needs to be through aerobic respiration. It cannot kick into lactic acid. Only one way. So that's why your brain is going to be the most susceptible part of your body when there is not enough oxygen or there is not enough glucose. Mitochondria provides that fuel for the nervous system. Okay? There are a lot of other things. We got endoplasmic reticulum. This is place that you produce the proteins. No problem. You got the gene. Gene has the instruction how to go in and make something. But if you are going to use that something in order to perform a function, you have to have the right shape. Right shape is just not going to be determined by the sequence, but you have to have enzymes in places such as the endoplasmic reticulum to fold it. Think about it. I have used this example before, but let me do it. If this key comes out and this key is going to be bent, it's not going to be able to open the door. What happens is that this key is going to go inside of this endoplasmic reticulum. There are enzymes that are going to make it straight. Now this key is going to be able to open the door. This is the way it's going to work. So you need the right shape of that protein. Most of those things are going to happen in here, endoplasmic reticulum, or another part, we call it the Golgi apparatus. It's going to happen there. And again, you're going to have a lot of other things, but we're going to talk about these things. How many types of cell do we have in our body? About 200. Okay? 200 from original zygote, when we had the sperm and the egg fertilize each other. You go through the embryogenesis and the division, ultimately what you are going to have, you're going to have 200 different cells with 200 different functions. Now, many times we have idea of the cells, something look like that, with a round nucleus and the round cell. Yes, you have many cells that are going to look like that, but not every cell is going to be like that. So I want you to remove this concept that always the nucleus has to be round. This is a neutrophil. This bluish part is the nucleus. So nucleus can become different shapes and forms. So don't have this concept that nucleus has to be always round, because it is not. Look at this one. Look at this cell. This is a neuron. Look at this cilia. This is the goblet cell. This is going to be this lining of the trachea. This is the lining of bladder. This is the transitional epithelium. This is smooth muscle. you can see this is sperm. You can see all these cells look differently. What is the reason for the different appearances of these cells? Different gene expressions. The genes that are going to be active in this cell are not necessarily the same genes that are active in this cell. Do they all have the same DNA? Yes, they do all have the same DNA, with the exception that sperm has half of that DNA. Okay, because you know the idea of sperm and egg, they're going to contain only half of the genetic material. Okay, so be aware of that. Keep these things. Look at the body as a whole. Don't look at the body separate things as a whole. So that's going to give you a better understanding what's going on. And this is the question that I always ask, but you already know the answer because we discussed it. If we are starting from the zygote, how is that we have 200 different cells with different functions? Is all going to be the fact that different cells are going to have different gene expression. I throw these things, this trivial information sometimes because it makes it fun. It is good to know these things. It makes you wiser when you understand these things. Let me tell you one of those trivial information that's going to make you wiser. Do you know when you're, what's the right word I'm using here? When your destiny has been determined. What is the moment that your destiny has been determined? I'm talking about science. I'm not talking about spirituality and those kind of things. I'm talking about real stuff. When your destiny is going to be determined. What by that I mean is that why should you have your mouth in here? Why shouldn't you have your mouth up your head? These are simple questions. Why my arms are going to be here? Why my arms are not going to come in my chest? When those things are going to be determined. Do you know that? You want to take a guess? Let me tell you that. What? The moment, this is cool, the moment that the sperm enters the egg, that's going to determine where your mouth is going to be, where the anus is going to be, where are your arms going to be developed. That moment, that point that the sperm enters is going to be the reference point. The gene expression that we're talking about is going to be determined right at that moment. And that's just amazing to me, okay? Because it's completely random. It's completely random, okay? It's pretty cool. These things are really cool when you understand how they work. Let's talk about the first one, the most important one, because living cells would not exist without a cell membrane. Cell membrane has a very unique property. It's semi-permeable. Let me explain to you what it means, semi-permeable. It means it lets certain things to go through, other things cannot go through. That's what it means. Now, what is the property of the cell membrane? Well, it's going to be primarily consist of phospholipids. Phospholipids are going to be very interesting molecules because they're going to have a very large tail that is going to be non-polar. They don't have a charge. But they're also going to have a head that is going to be polar. That's usually going to be phosphate that has charge. So when you are getting, if I be able to get a bunch of these molecules, these phospholipids, put it in a container of water and shake that container of water. I used to do this experiment because I had all the tools when I was teaching before. I don't have it in here to show you. I shake this thing, all these phospholipids are going to line up together. They're going to line up together. It's fascinating because this non-polar is not going to be dissolved in water. It's not like sugar that is going to be dissolved. It's like oil in the water. What's going to happen here is that these phospholipids, the tails are going to point to each other. The heads that are going to have charge are going to point outward toward the water. Okay? This is going to make this membrane a very important component of the cell because it's going to be semi-permeable. What does it mean? It means that if you have a sodium up here, remember it's a sodium ion, has a charge. Do you think it's going to be able to go through this membrane? No. It's fine up here. But this big tail is going to be non-polar. Non-polar molecules do not like polar molecules. They're not going to allow them to go through. Now sometimes you have a polar molecule that is very small in size, very small in size. To some extent can go in and out of the cell, like water. Water is very small and it's polar. However, it's able to go through because it's very, very small. But any other molecules, sodium chloride with ions, are not going to be able to go through this cell membrane. But what's going to happen if you need it? What happens if you need sodium inside of the cell? You got to have a mean in order to be able to get that sodium into the cell, and we do have that. Everything goes back to proteins. You got to have a protein that's going to act like a gate and allow the sodium to go through. We'll talk about that in a second. But this property is going to be very important. Now, think about it this way. Let your imagination go wild in here. Because your membrane, your cell's membrane are not set in concrete. If I am going to be a phospholipid, and there is going to be, of course, my legs are going to be the non-polar tail, my head is going to be the polar tail, and there is going to be somebody just like me, but their feet is going to be upward and they're in the floor. We are standing like this. When you're looking at the cell membrane, it's not that we are standing here and not moving. I might be here this second, 2 minutes later I might be here. 2 minutes later I might be here. You might be there. It's completely changing. We call that fluid mosaic model. It's not in cement. Things are moving. Continuously they're moving. But there is not going to be a gap in between. There is not going to be a gap in between, so things go from outside to inside without control. But there is going to be a continuous movement of these phospholipids. But when you are looking at this cell membrane, what you're going to be able to see in here, you see these big structures are going to be sticking out into the membrane. Some of them are not going to be sticking out all the way. Some of them are going to go all the way through. Some of them are going to be only on the outside. These are going to be all proteins. These are the proteins. You remember that I talked, for example, I told you that sodium has to go in. How is going to be able to do? With the help of proteins such as this one. You guys have done your blood types. You know your type O, B, whatever your blood type is. What does it mean? It means on the red blood cell membranes, you got one of these proteins that's going to be sticking like that, is going to make you type A. If you don't have this branch in here, you're going to become type B. If you have both branches, you're going to become type AB. If you don't have any one of these branches, you're going to become type O. It gives identity to your cells. Why people, when they get organ transplant, why somebody gets a kidney transplant and they have to be on immunosuppressant drugs such as cyclosporine for the rest of their lives? Because of the fact that the pattern of these information on their cells is not going to be recognized by their own immune cells. Immune cells are going to think they are foreign, and they're going to attack and destroy that transplanted organ, and it's going to be rejected. The transplant is going to be rejected because of these identities. That's why usually before the transplantation, we have to make sure that at least there is a similarity between those markers on the cells of the donor and the recipients. Okay, there's a lot going on in here with the cell membrane, but as we come to it, I'm going to describe every one of them in a little bit greater detail. We go through this. Gatekeepers. Going back to the sodium and potassium. So remember, what's that question? What was?
I was just double checking order rejection to MHC1 and MHC2.
That's it, because those are MHC. What Vinny is talking about a major histocompatibility complex. These are genes. You got 6 genes. They are going to be on chromosome number 6, and they are able to produce different things. I have my, for example, type 1, 2. Vinny might have type 3, 4. Somebody else might have 1 and 6. These are the genes that determine my immune response. Okay? So yes, that's exactly what they're going to go out and use in order to match the recipient and the person who is going to donate the organs. They check for those genes to make sure there's compatibility. Yeah. We said that some cells, some structures, some molecules are needed inside of the cell. You cannot get them inside of the cell because they're polar. Or sometimes things need to go outside of the cell and they cannot get in because they are polar. How are we going to be able to do it? You can see a very good example up here. This is sodium-potassium transporters. no, actually this is not it. This is for acetylcholine. This is going to be, so let me see how much you guys remember from your physiology. Let me see how much you're going to remember that. If I want to go ahead and contract my muscles, my brain tells hey, you're going to go ahead and contract this muscle, sends the information to my spinal cord, and from spinal cord, the motor neurons are going to go to the muscle that is required for the movement of my fingers and hands. But whenever that neurons, the motor neurons are activated, what happens between the neuron and the muscle? How does the information from the neuron is going to be translated into the muscle? Hey, contract. Don't relax, contract. You contract this one, contract that one, and contract this one 80%, contract that one 60% in order to be able to get something like a movement. Not myosin, before myosin. What's that?
Calcium.
Not calcium yet. You are inside of the cell. I'm saying between the neuron and... Alright, you guys don't remember it. What was the neurotransmitter involved?
Sending a signal.
Neurotransmitter that is going to be transferring the information from the neuron to the muscle. You guys don't remember acetylcholine?
Yeah.
Alright, acetylcholine. So the story about acetylcholine is like this. For the muscle, in order to go ahead and contract, you got to generate action potential. What is action potential? I think I told you this class before. Every cell in your body, the membrane has potential. What does it mean? Can anybody tell me what does it mean when I say this cell, the membrane has membrane potential? What does it mean? Just give me a simple word, layperson's language, what it means, then we get into details. Means there is a difference in the charge concentration inside versus outside. So relatively outside is going to be more positive charges versus negative charges on the inside. Okay? That membrane is going to have potential. Okay? So what's going to happen here is that for the muscle to contract, you got to generate action potential, which means that for a short period of time, you got to change this potential. So inside is going to be positive, outside is going to be negative. How would you be able to do that in the body? You got to be able to move ions in and out. So when I wanted to move my finger, my muscles, what I did that my motor neuron released a neurotransmitter called acetylcholine. Acetylcholine at the synaptic cleft. Remember the synapse, the muscle? Acetylcholine is going to be released from the motor neurons and is going to have, this is going to be the muscle membrane. There is going to be a gatekeeper on the muscle membrane that is always closed unless you are going to have acetylcholine coming and sitting on the spot that belongs to it, and that's going to be the key that's going to open up the gate. So then when you are going to go and open up this gate, you have more concentration of the sodium outside of the cell than inside of the cell. Where does the sodium is going to go? It's going to go inside. When it goes inside of the cell has a negative potential, is more negative than the outside. What is the charge of sodium? Positive. Is inside is going to become more positive, outside is going to be relatively more negative, and that's what's going to allow the action potential, and that's what's going to allow me to move my fingers. Very simple concept, okay? But you would not be able to do it without acetylcholine sitting there. Has anybody seen any, well, probably you haven't seen. Have you ever seen a picture of a person who is dead from Mycobacterium tetani infection? You know, whenever you get tetanus vaccination, why do you get tetanus vaccination? Because in dirt there is this bacterium, Mycobacterium tetani, that whenever you go and hurt yourself and get it in your skin, what's going to be able to do is going to produce a toxin. Have anybody seen any picture of the person who died from Mycobacterium tetani? They are like this. And they usually die because of the fact that because their muscle contracts so severely, that's going to break their spinal cord and they're going to die like this because they cannot breathe anymore. Why? Because Mycobacterium tetani is going to produce a toxin that's going to act as acetylcholine. It's going to sit in here, not on finger only, all over the body. And the difference is that acetylcholine is going to sit. It is an enzyme called acetylcholine esterase. It's going to come and cut it off, and it's going to break it down, so everything is going to close down. That toxin is going to sit, and it's not going to go anywhere. So the channel is always going to stay open, and the muscles, entire body are going to be contracting, and that's what's going to cause the tetanus and the death of, because of that bacterial toxin. You have a lot of these gatekeepers. This is one of them. I was talking to somebody, I don't remember who it was. I know 45 different gatekeepers just for sodium in your body. And that's what I know. I know that there's going to be more. 45 different ones. You got different ones for potassium. You got some that are going to let the sodium in, potassium out. You got something that's going to let the potassium out, sodium in. You got some of them that's going to let the hydrogen ion in, sodium out. There are a lot of different ones. But the principle is not going to be different. You got a ligand, such as in this case acetylcholine, the key, that usually the gatekeeper is going to be closed unless that ligand is going to be an opening it up. And what's going to happen is that things are going to happen, whatever that ligand is going to be doing. All right? That's the gatekeeper. These are all proteins. Another one is cystic fibrosis. What do you think cystic fibrosis is going to be? What is the disease called cystic fibrosis? What do you know about it? Yes, you produce, you would see it in the respiratory system, but you're really going to have problem all over your body. We just have the respiratory system because that's going to have the major symptom. You're going to have a problem with pancreas, you're going to have problem with liver, but primarily it's going to be your respiratory system, your bronchi, your lungs. You know what it is? There is a channel, one of those gatekeepers for chloride ion, for Cl. There's a genetic mutation in the channel for chloride ion. Every symptom that you see in a cystic fibrosis patient is because of that mutation in that channel. So that previous channel, gatekeeper that I showed you, it didn't work right. It was not for sodium, it was for chloride. What's the outcome? You have cystic fibrosis, okay? Now you have also these proteins that are going to have different features. Sometimes you're going to have lipoproteins, a protein with a lipid on it. Sometimes you're going to have glycoprotein, a carbohydrate with a protein on it. Most of these things are going to happen inside what we call Golgi apparatus inside of the cell. That they're going to, this protein came with the DNA, but this sugar part, this glycoprotein that was added to this protein was added later on in the Golgi apparatus or sometimes the endoplasmic reticulum, but mostly at Golgi. Okay? It's going to have marker. It's going to have what I just told you about your blood type. Different blood types are going to be exactly as this. This is the way that your blood types work. If you don't have any of these carbohydrates, you're type O. If you have this branch and this branch, you're going to be type AB. If you have this one, you're going to be type B. If you have this one, you're going to be type A. That's it. The identity of your cells. But of course, you have more than 50 different types of these markers on your, for those of you who are going to take blood bank, you don't have just 4 blood types. You got a lot more. Every cell in your body has these markers. When you are having an infection, ask yourself these questions because you got to think about these things. I know that in this heat it's very difficult to think, but what happens is that you got to think about these things. When your immune cells are trying to attack a bacteria or a virus or something that's going to be in the wrong place, how do they differentiate between your cells and enemy cells? How do you differentiate between self and non-self? They check for these. Why it is so difficult to be able to go ahead and deal with viruses? Why is this Zika virus that's causing havoc in South America causing so much problem? Why can't we just go ahead and treat them? Well, because the fact is that these viruses are really tricky because they get inside of our cells. When they get inside of the cell, this is the cell, this is the virus, this is virus that's infecting the cell. When the immune cells are going to come, they're not going to see the virus, they're going to see that, hey, this is my cell. Go on to the next one. And this virus is able to replicate. And then it's going to let go, and 1 million new viruses are going to come in. It all makes sense as long as you understand the basic structure of the human cell. Everything, the story starts at the human cell. Now, these are going to be the receptor models. It sounds silly, the example that I'm going to go ahead and give you, but again, think about what I'm trying to tell you in here. Try to grasp the concept. Most of the boys, when they are going through the teenage years, they're, you know, hitting through the puberty. Before the puberty, skin is soft and nice and beautiful. They hit the puberty, all of a sudden, there are a lot of changes that are going to happen. One of those changes is that they're going to start growing hair on their faces. Have you ever asked yourself, why don't they start growing hair on their forehead? Why? Is this a skin? So is this a skin? How is that you are able to make that difference? Why there is something happen? Why when you are producing, for example, let me come up, cortisol, a hormone. Why that hormone is going to be able to affect certain places in your body, but not other places in your body? Because it's going to be a hormone. Hormones are going to travel with blood. Every place that blood goes, hormones go. But some cells are affected, some cells are not affected. Why? It all depends on the receptor. If you have a receptor for a hormone, you will respond to that hormone. If you don't have a receptor for a hormone, you're not going to respond to that hormone. And the genetic expression, that's going to be different. So the hair that are going to be, the follicles that are going to be on the face, in the face area, they're going to have a receptor for testosterone. They're going to respond to it, and they're going to cause a growth in hair. But the forehead, they don't express it, even though they are both skin cells, so they're not going to respond to testosterone. A simple concept. But that would not happen without these proteins that act as a receptor. A cell will respond to something if they have the receptor. They're not going to respond to it if they don't have a receptor. So why am I telling you these things? Because that's why you are here. I hope that you are here to understand about the human diseases and how you would be able to identify and detect these things. Get a fundamental understanding. This is going to be familial hypercholesterolemia. What is that? Look at the position. These are all fat deposits. This person, this is a 40-year-old female with that. You can see how it looks. It's a terrible disease. Most people will die. They're not going to even see this 40-year-old was a lucky one, because usually by the 30s they're dead. They are going to have a heart attack, they're going to die, because of the blood vessels are going to be all occluded. What is happening here? Well, we need cholesterol, and I'm going to go ahead and emphasize this one more time. Cholesterol is not a bad thing for those of you who think that cholesterol is a bad thing. Cholesterol is a very good thing. Cholesterol is going to be a very important thing for your body, but goes back to everything else. Homeostasis. Too much of anything is not going to be good. As long as you've got the right amount of cholesterol, it's going to be necessary for survival. People who are having their cholesterol going very, very low, below 100, they develop schizophrenia and they will die. So don't think that, you know, the reason I'm telling you, and you got to think about these things in a bit more in profound way. You go to grocery store, what you going to see? Fat-free this, fat-free that. What the heck is that? What is all about fat-free this and fat-free that? Is it healthy? It is very unhealthy to eat those things. You eat things with fat in it, but not too much. The right amount. 20% of your calories should come from fat, not 80%. Okay? That's the problem that we have to deal with. Not to use people's ignorance and sell them products while you're selling something that's not going to, or these sweeteners. If you're not diabetic, don't drink these, diet sodas and things like that. Those kind are good for people who are diabetic. Otherwise, all these sweeteners are going to have a negative effect on your body. And these things are coming out. After 20 years, people are developing so many different kinds of gastrointestinal diseases because of they have been using this kind of sweeteners. Now we are understanding, we are saying, oh, you should not do it now. Okay? Educate yourself. This is the same story. This is genetic mutation. So what happens here is that these people, so let me just tell you a quick review about cholesterol. So cholesterol is a lipid, right? Lipids cannot travel in plasma. Why? Because plasma is going to be all water and lipids don't mix with water. Okay? So you have to have a carrier. A carrier in the liver is going to be responsible to take the cholesterol and take it to the tissue. A carrier in the tissue is going to be responsible to get the cholesterol, bring it back to the liver, and the liver cells are going to get it out, put it in the bile, and they're going to put it in your intestine and you're going to get rid of it. This is the cycle. From the liver to the tissue, from the tissue to the liver. Now the problem is that the one that's going to go from the tissue, from the liver to the tissue is low density lipoprotein. Is it protein? Is it carrier protein? Okay? And the one that's going to be getting the cholesterol from the tissue, take it back to the liver so you can dispose it, is called high density lipoprotein. It's a protein carrying cholesterol. Now these people have a genetic mutation in their receptor. So the way it is that this is the cell, this is the receptor for low density lipoprotein, LDL. So cholesterol comes in and sits on that receptor, and then things are going to happen. Cholesterol is going to be moved into the cell. These people have a mutation in that receptor. So the cholesterol cannot get inside of the cell effectively. So what's going to happen to that cholesterol? It's going to start depositing under the skin. It's not only under the skin. If you look at this person's artery, it's going to be filthy. It's going to be a lot of plaque in it. So that's what happens. A simple genetic mutation. One of those A's and T's and G's and C's can go ahead and be mutated, and this would be the outcome. Okay? Usually it's going to be much more complex. Usually we have more than one mutation that causes a problem like this. For this case, one single mutation can cause that. The nucleus, again, in eukaryotes is going to be the center, the place that you are going to have the majority, 99.9% of your DNA. Is going to be the place that you will maintain this information. And you will be able to use this information as you need it. There is this concept that we will refer to it in a second, but let me just go ahead and tell you. The basic principle of biology, the central dogma of biology, they call it, is the concept that most of you know, and that's the concept that you have DNA in the nucleus. In the nucleus is going to be transcribed into RNA. This process is called transcription. And the RNA is going to come out of the nucleus in the cytoplasm, and it's going to be translated by the process translation and becomes protein. This is the central dogma of biology. DNA, RNA, protein. Transcription and translation. Transcription is going to occur inside of the nucleus. Translation is going to happen in the cytoplasm. Okay? You can ask yourself these questions. This is where the learning is going to happen. How on earth this RNA is able to come out? How is that possible? Well, you have nuclear pores. A nuclear membrane has a lot of pores that will allow RNA to go through, but they're not necessarily going to allow everything to go through. Okay? Those nuclear pores are going to be very important. Why? What happens if there is something wrong with one of these nuclear pores? You develop devastating diseases. How many of you know what progeria is? Progeria. Have you heard of progeria? Oh, I have a picture I will show you. Progeria is one of those cases. One of these proteins that make up the nucleopore has a mutation. It's called lamin A. Has a mutation in it. It's going to cause progeria. It's one protein. Okay? So it's going to be very important because the nucleus cannot divide the way that it should. The person is going to age. So a 5-year-old is going to look like a 20-year-old when it comes to their physics, their body. They're not going to grow, of course. They're going to lose their hair completely. They're not going to have any hair on their body. They're going to look very aged. And they're going to die usually by the age of 20 or 30. They're going to die. They're not going to make it longer than that. Nucleolus is going to be inside of the nucleus, and I will show you other things about nucleolus. This is not a good image, but it's going to be inside of the nucleus. Nucleolus is the place that you make RNA. That's where RNA is going to be synthesized inside of the cells, inside of the nucleolus of the nucleus. And of course, chromosomes are going to be long line of nucleotides. Different organisms have different number of chromosomes. Humans have 23 pairs, which means 46 chromosomes. Just because you have more chromosomes, that doesn't make you more intelligent. Because rice plant has 72. Okay? So that's not, rice plant, last time I checked, is not going to be smarter than us. So it's all going to be what function we're going to do. Okay? So number of chromosomes has nothing to do with anything. It's just humans have 46. But there are conditions in which we might have 45, we might have 47, we might even have 48. But when we come to it, I will discuss those conditions for you. Okay, these are the neutrophils. I want to go ahead and start training you that things may look different, even though they're going to be the same thing. Both of these cells are going to be neutrophils. They have been stained with right stain, so anything that's going to be deep basophilic is going to be the nucleus. Because the DNA, the stain is going to be basic, is going to bind to the DNA deoxyribonucleic acid, and it's going to stain it very dark blue. Look at this. The nucleus in here and the nucleus in here. They're different, aren't they? It's okay. It happens. I can tell you one thing right now. This is a younger cell than this cell. You will learn these things. So when you're looking at your blood smears and you have to identify the different white cells, when it comes to this one, you're not going to call that segmented neutrophil. You're going to call that band neutrophil. Band neutrophil is important because you know why? Usually I would expect about less than 3% or 3 to 4% of your white blood cells to be band. Now I am looking at your blood smear and I see 20% band. How would you be able to interpret that? What does that tell you? There is something going on that your bone marrow is kicking out these white blood cells out of the bone marrow into the blood in order to be able to deal with the situation. Okay? So that's going to be very important information. And you obtain that just by looking at it and paying attention to these cells. And again, forget about the fact that the nucleus always has to be round. It's not round. All right, humans have 46 chromosomes. You can see that, I cannot get it down, but you can see some of these chromosomes up there. Okay, this is going to be some, I can tell you because I can see the 23rd pair is going to be a female on this side, is going to be a male on that side. Each chromosome has a number. Do you know how you number them?
Based on size.
Based on the size. The longest chromosome is going to be number 1. You got one from your mother, you got one from father. When you are looking at this chromosome, let's just focus on number 1. If you, this is not going to be, I'm just trying to make a point in here. If you, listen up, this is very important if you don't know it. If in this area of chromosome number 1 that she got from the father's side, you got a gene for your hair color, on the mother's side, on the same spot, you will have a gene for the hair color. It might not be the same color, it might be a different color, but it's going to be for the hair color. Okay? So if you are going to get the brown gene from your father and brown gene from your mother, you're going to become brown hair. If you're going to get blue from your father and brown from your mother, you're still going to be brown. Because of the fact that brown is going to be dominant over blue. But what I'm trying to tell you that we have 25,000 genes in our DNA. They are going to be distributed on these chromosomes, but they're going to have a counterpart on the other side. So you have 2 different types of diseases. Get that down so you understand when I'm talking about the autosomal recessive and autosomal dominant versus sex linked. When I am talking about recessive versus dominant, this is what I'm talking about. If a disease is going to be caused by having one mutant gene, it's going to be dominant. if for that disease to show its characteristic, the word is phenotype, you are going to have a recessive one, you need to have 2 defective genes. The mother gives a defective gene, the father gives a defective gene, and then the person is going to develop it. That's a recessive trait, okay? If you are going to be able to show the trait, to show the phenotype or the disease or characteristic with 1 gene, that's going to be dominant. So when we are talking about diseases such as Huntington disease, on chromosome number 4, halfway down here, there's a gene called Huntington gene. If you have 1 mutation, now you're not going to, it's not single mutation, it's a addition mutation. You're going to add an extra chromosome to that single gene. If you have 1, you will develop Huntington disease. There is no if or but about it, it's just when. Okay? But if you are having cystic fibrosis, you got 1 mutant cystic fibrosis gene for the chloride channel, you will not develop cystic fibrosis. You will be the carrier. You can have a child if you marry somebody else. If you marry somebody who is having a defective gene, you can have a child with cystic fibrosis. Because if that person gives a defective gene, and you give the defective gene to the child, that child is going to be homozygous. Homozygous means they are going to be the same. Heterozygous means one of them is going to be one thing, and the other one is going to be another thing. Everything is going to go down here. It's going to come down to the DNA. Okay? Yes, you can develop some kind of anatomical trauma and die. But when you're talking about disease development, it's all going to come down to the DNA. It's all going to come down to these chromosomes. And when you're looking at the chromosome, I want you to develop an understanding what they are actually look like. So this is the cartoon of it here, you can see. But if you want to go in and unfold it and a little bit simplify it, if I am walking, this is the beginning of this chromosome number 1. If I am walking in the chromosome number 1, I am going to go and continue walking until I go all the way to the end of it. Because chromosomes in humans, in eukaryotes, are going to be linear molecules. So if I am having a chromosome in here, if I start walking down here and go, I'm going to end up down here. That's not true about mitochondrial DNA. That's not true about prokaryotic DNA. Because their chromosome is circular. If I start in here and walk on that DNA, I'm going to end up where I started. Okay? So be aware of these. These are just what? If I am having this imaginary microscope that is enabling me to magnify this unlimited number, what am I going to be able to see ultimately if I'm looking at this chromosome? A's, T's, G's, C's associated with proteins, and that's what's going to be. There's nothing fancy about it. The order, the sequence of these A's and T's and C's and G's is going to determine what's going to be the outcome. But that's what you're going to be able to see on the chromosomes. And these are the chromosomes. This is the electron microscope image. All these dark parts, you are going to go in and have dark areas inside the nuclear membrane is going to be DNA. Those are genetic material. Some places you're going to have darker. Some places they're going to be lighter. Does anybody know what these are? What's that? No. See? Yeah. What happens is that I told you got 25,000, 24,000. I have seen many numbers: 24,000, 500, 25,000. Some place along that you got that many genes in every cell, right? In every cell. But how many genes I told you on average are going to be active in every cell? Okay, can I borrow this? If you are having, this is being a DNA. This is being a gene. For this gene to be able to make the product, enzymes have to sit on it, and they have to go in and read it in order to do the transcription, and RNA is made and sent to the cytoplasm for the proteins to be made. You understand what I just said, right? But do you think that enzymes are going to be able to reach this when I like this? This is the way that you're going to deactivate the genes. By twisting them in a way that the enzymes are not going to be able to sit on them. And this is the way that some are going to be darker because these are the folded DNA. Some are called euchromatin. They're going to be opened up because they are going to be red. Okay? And that's going to be different. It's going to be different for each cell. The blue in here, now This is pseudocolored, so the red is going to be cytoplasm. You got two centrosomes, mitotic fibers. I'm going to talk about those in this lecture. And the blue are going to be the chromosomes. These are going to be the chromosomes that are going to be divided at the time of mitosis and meiosis. Last time, a chromosome is going to be a sequence of A's, T's, G's, and C's. There are going to be parts of that chromosome that are going to code for different proteins. You have two genes here, gene number 1, gene number 2. Gene number 1 is going to produce a product that is going to give you a different eye color. Gene number 2 is going to be able to make a product that's going to give you a different hair color. And the same thing is going to be for blood type. Same thing is going to be for different type of enzymes. But the idea is that different genes are going to make different proteins, and different proteins are going to have different functions. This is progeria, if you didn't know what progeria is. This kid, I think he's 9 years old, and he has one genetic mutation on one of those proteins that make up the nuclear pore. That nuclear pore that allows the stability of the nucleus, that nuclear pore that was allowing the RNA go through, there's a mutation in one of them. And this is the outcome. Okay? Large head, losing all the hair. They are fully intelligent. They are, even if they have done a study that look at the level of intelligence, they are even more intelligent than you and I. Okay? In that sense. But they will die. They will not make it more than the age of 20. Usually they die in their teens because of that single mutation in that single protein. This is what happens when things go awry with this sequence of the DNA. The protein that's going to be involved is going to be lamin A. Get to know this image because you're going to get to know these cells very well by the end of this semester. These are a group of cells, we call them blasts. Don't worry about what it means right now, but remember the name, blasts. And when we are talking about nucleolus, we are talking about nucleolus. That's one of the main characteristics of the blasts. They have prominent nucleolus. Okay? But the bottom line, you can see the nucleus boundary in here, you can see cytoplasm boundary in there. The size and the ratio is going to differ. People are going to come, you guys are going to, hopefully you're going to come to me in the middle of the semester and say, Dr. Nasser, I cannot identify this cell from that cell. I don't know whether or not this is immature or mature. What would you tell me? I would tell you that it's very simple. Number 1, look at the size of the cell. How can I go ahead and determine the size of the cell? Look at the RBCs. There is always something there that's going to give you a hint what the size is. Look at the appearance of things such as the nucleolus. You're not going to have nucleolus, for example, in neutrophils. You're not going to have it in monocytes. You will have it in blasts. You will have other cells as well, but you're going to learn those. Look at the ratio of the nucleus to cytoplasm, because that ratio is going to change. Look at the cytoplasm color. These are the hints that you got to go and use in order to identify it. But you got to pay attention to it. If you don't pay attention to it, you're not going to be able to do it, right? But it's simple. It's not hard. With more practice, you're going to get better. Cytosol. So cytoplasm and cytosol, what's the difference? Anybody know what's the difference? Cytoplasm, cytosol? So inside of the cytoplasm, you got a lot of things we call organelles. If you remove the organelles and you just leave that fluid, you leave that white part of the egg, that's what the cytoplasm actually going to look like. And the consistency of the cytoplasm or cytosol is going to be exactly the same way that the white part of the egg is going to be uncooked. When you cook it, that's going to be different. Okay? It's semi-liquid, but all these chemical reactions are going to be happening in here. Can anybody think of a chemical reaction that happens in cytoplasm? Glycolysis. Glycolysis. That's a good one. Glycolysis, exactly. They're all going to happen in here. Not in here. Okay? In here. In cytosol, a lot of enzymatic reaction. You also do a lot of storage. For example, you would be able to, adipocytes are fat cells. Adipocytes are fat cells. Where do you think the fats is going to be stored? Where do you think the lipids are going to be stored? In the cytosol. Inside these vacuoles, you're going to be able to store the lipids. So if you see a big guy who has been losing weight over 3-4 months, and they have shed 30, 40, 50 pounds, if you want to really know about this, this is going to be the situation. They still have the same number of fat cells in their body, okay? But their fat cells are not going to have the same content of lipids. They have lost the content of those lipids inside the vacuoles, and this is where the weight loss has come from. Okay, at least the healthy weight loss should come from there. I told you about central dogma. Okay, organelles. So you got different organelles in the cytoplasm of the cell. Some organelles have their own membrane, but it's not going to be any different than the plasma membrane, which means it's going to be phospholipid bilayer and it's going to have the same characteristic, which is going to be very important. You're going to have some that are going to have membranes. You're going to have some that are going to have double membrane. They're going to have two membranes, such as mitochondria. Ribosomes. Ribosomes don't have membrane. Ribosomes are hybrid molecules. What are they made of? Does anybody know what ribosomes are made of? What are the two major components of it?
Protein.
That's right. RNA, we call it ribosomal RNA, and protein. Okay? There's a protein and this RNA going to come together, and the hybrid molecule is called ribosomes. And they are going to be able to do what? They are going to be able to, remember when I told you, you got a gene. Let's put this in perspective. If you don't understand what I just said, I'm going to say, I want you to raise your hand and ask questions right now, before we go any further. You got a gene in the nucleus. That gene needs to be expressed, which means an enzyme is going to sit and read it. What is it going to make out of this by the process of transcription? RNA. RNA is going to come out of the nucleus, get into cytoplasm. When it gets into the cytoplasm, it's going to be captured by ribosomes. And the ribosomes are going to read the sequence of RNA. They're going to say, okay, A, U, G. This triplet is called a codon. They're going to read this sequence of RNA in triplets. Every 3 of them are going to be able to be read. A, U, G is methionine. They're going to get an amino acid, stick it up here, and they're going to read the next 3. Leucine, another amino acid. They're going to bond them together by a peptide bond, and that's the way that the polypeptide chain is going to expand. What is that? A polypeptide, another name for a polypeptide chain is what?
Protein.
Protein. Now that protein might not be ready to do what it's supposed to do because it doesn't have the right shape. It does have the right sequence of amino acid, but the shape, there's a problem with it. That's the time it's going to go to endoplasmic reticulum and Golgi apparatus, and it's going to be folded, and it's going to be cut, and it's going to be beaten up in order to get the right shape at the end. Okay? Everybody understood that, right? So those are the ribosomes. There is, in this image, there is a chain of RNA. You don't see the chain of RNA because it's very, very narrow. But every one of these black dots that you see there is a ribosomal complex. So you can see, you got one chain, you got hundreds of ribosomes reading the same chain, like an assembly line. So from one RNA chain, you would be able to make hundreds of copies of that protein from one single RNA. Okay? Endoplasmic reticulum. Again, it's going to be double. It's going to be membrane. It's going to be a sac. It's going to be like a puzzle. It's going to be a labyrinth adjacent to this area. This is the nucleus. This is going to be the endoplasmic reticulum. It's double. It has its own membrane. Inside of it, things are going to be different than outside. So inside of the endoplasmic reticulum, you're going to have much more calcium, 1,000 times more calcium than you're going to be able to find in the cytoplasm. When I was giving an example about muscle contraction, Some of you said calcium. That calcium that you were thinking about muscle contraction, that is going to come from endoplasmic reticulum. It's not going to come from outside of the cell, at least for the skeletal muscle. It's going to come from the endoplasmic reticulum. Okay? So what's going to happen here, a lot of things are going to happen. You have a lot of different enzymes. So when you make that protein doesn't have the right shape. It's going to be put inside of the endoplasmic reticulum, and the endoplasmic reticulum enzymes are going to fold it the right way, and they're going to process it further. So we are going to have two different types of endoplasmic reticulum. One type is going to be called, the rough endoplasmic reticulum. The word roughness is going to come with ribosomes are sitting on it. Look at this image down here. These dots that you see in here, those are ribosomes sitting on the endoplasmic reticulum. Why? Okay, I am going to make this protein. But I know that the product of this protein when it's produced is not going to have the right shape. This protein has to be made in endoplasmic reticulum. If I was going to make a protein with these ribosomes, that I was going to use it exactly as it is, you wouldn't make it in here. You will make it in the cytoplasm. Okay? You will make it in the cytoplasm. If a protein does not need to be processed, change its shape, things added to it, you will make it in the cytoplasm without anything, and you're going to let them go after the business. But if they need to be processed, most of the proteins are going to need to be processed. They're going to be happening in the endoplasmic reticulum. Okay? You also got a smooth endoplasmic reticulum, the one that doesn't have ribosomes with it. That endoplasmic reticulum is going to have many different functions. One of them is that anytime your body makes steroids. Testosterone is a steroid. Cortisol is a steroid. Aldosterone is a steroid. Vitamin D is a steroid. All those things, when they are being made, they are made always in the smooth endoplasmic reticulum. Okay? So if you look at your adrenal gland, you have a lot of steroid hormones there. You got DHEA, you got aldosterone there, you got cortisol there. They are all going to be produced in the cells there, and those cells, when you look at them on the microscope, they got a very elaborate smooth endoplasmic reticulum. Another cell in your body that's going to have a lot of smooth endoplasmic reticulum is going to be your liver cells. Because liver is the place that you're going to detoxify toxins. You're going to get, whenever you are eating something that has toxic characteristic, that material is all going to be broken down in the smooth endoplasmic reticulum of your liver cells, hepatocytes. Okay? So detoxifying, making lipids, making steroids, they are all going to, and having storage for the calcium, they are all going to be done in the smooth endoplasmic reticulum. Protein processing, modification, endoplasmic reticulum, the rough one, not the smooth one. Golgi apparatus. So this is a beautiful image of it, but let me go to this cartoon down here to explain to you what happens. Okay? Pay attention up here, because this basically puts everything together. We are talking about rough endoplasmic reticulum up here. This is a ribosome that's sitting on it. That's why I know it's rough. It had captured RNA and is translating the RNA into a protein, but the protein is going to be released in the cistern or inside the endoplasmic reticulum. But this protein doesn't have the right configuration. Enzymes that are going to be in here are going to twist it and fold it, and they are going to get it ready. And when it's ready, at the end of this endoplasmic reticulum, it's going to be put in one of these sacs, and these sacs are going to bud off. And they're going to be released in the cytoplasm. And the cytoplasm, they're going to travel like this. They're going to travel, and they're going to come to the Golgi apparatus. They're going to fuse with Golgi apparatus, and that's not the miracle, because this is phospholipid. The membrane of the Golgi apparatus is phospholipid. When you have a pot of water, you have 2 drops of oil in it. What happens for these 2 drops of oil when they come together? They fuse. They fuse, right? Same thing is happening in here. They fuse, and this vesicle is going to release this component into the Golgi apparatus. Now you got enzymes. You can see there are stacks of Golgi. In every stack, you got a different group of enzymes. And they know that, hey, if this protein comes in, I got to chop off 2 amino acids from this side. I got to chop off 3 amino acids from that side. And they process it one step, and they bud it off. Got to go to the next sac. More modification, gonna go to next sac. Gonna go to next sac. Ultimately, when the protein is ready, they're gonna put it in the vesicles and they're gonna release it. There are a couple of possibilities. What's gonna happen to that protein? Usually, when the proteins are being processed this way, they're gonna be released outside of the cell. Like hormones, you need to release them, so they're gonna go do different things. They're gonna come to the membrane. What's gonna happen when you get to membrane? They're gonna fuse with the membrane, because membrane is gonna be phospholipid as well, right? They're gonna go then to the blood, and blood is gonna go and take it up. Sometimes it's not gonna happen like that. For example, lysosomes, they are produced like this. Inside these vesicles, if it's gonna become lysosomes, you're gonna have 40, 50 different enzymes. Enzymes that are gonna be very harsh enzymes. Inside of the lysosomes, the pH is going to be about 4.0, okay, 4.5, 4.0, very acidic. In the cytoplasm, pH is going to be 7.4. So it is going to be very important to make sure that the enzymes that are going to be in here, they don't get their hold on the cytoplasm, because they're going to destroy everything. And that's what happens when the cell dies. When the cell dies, it lets all these enzymes out. And they're going to start chewing off everything in their path. So the idea is that if there is going to be lysosomes, it's going to be inside these vesicles, and it's going to be released in the cytoplasm. It's not going to come out. So it all depends on what is the purpose of those proteins that are going to be inside of the vesicles. Is it going to go outside of the cell? Is it going to stay in the cell? Okay. I'm going to give you 5 minutes. If you want to take a break, take a break for 5 minutes. Come back. I'm going to start right at 2:20. Okay. And if you look at this image down here, it's going to tell you the entire story. From the endoplasmic reticulum to Golgi apparatus to release the product into the outside interstitial space. Okay. Is there any questions so far? So this is the story about lysosomes. The origin of lysosomes is going to be Golgi apparatus. That's the way they're going to be produced. But then they're not released outside of the cell. So what's going to happen here is that you have a lot of things are going to be made, things are going to be broken down, things are going to be recycled. So there's a lot of turnaround in the production of these mitochondria, ribosomes, things like that are going to be continuously made and broken down and recycled. In here, you can have the bacteria, or you can have organelles that are old. In this case, we are having a mitochondria. It has done its job. Now it needs to be recycled. How is it going to be recycled? It's going to be done by lysosomes. What's going to happen here is that mitochondria is going to be put inside of a vesicle. That vesicle is going to come and fuse with this vesicle that contains the enzymes inside of the lysosome, and these enzymes are going to start to break down the mitochondria. They're going to break down the proteins to amino acids, carbohydrates to sugars, and the lipids to fatty acid and glycerol. And if there's a DNA there, they're going to break it down to nucleic acid, A's and T's and G's and C's, and they're going to be using up in other things as time goes by. Okay? So this is the way they do it. And it's very important because you never expose these enzymes to the cell itself. Because if they are exposed, they're going to destroy the cell. They're going to kill the cell by something we call necrotic death. They're going to destroy the cell. They're going to cause inflammation. And we'll talk about that later on a little bit more. You have a lot of enzymes, as I told you, inside the lysosomes. One of these enzymes, I just want to give you one example so you would be able to apply things that you are learning. It's going to be so this is the same idea of lysosome production. So you can see that this is, let's imagine this is going to be a neutrophil phagocytic cell. These are bacteria. They are going to be phagocytosed. They're going to come in. You got it here inside the vesicles. It's not going to hurt the cell. You got the lysosomes with the enzymes in here. They're going to come. They're going to fuse, and the enzymes are going to get busy and pick up the business in here. Same concept. Makes complete sense. This child is a 2-year-old kid. She was not born deaf. She was born actually normal, complete normal, until the 3 months it starts symptoms showing up. Blind, deaf,
cannot move, cannot eat, cannot do anything, and she will die within a year or two. Okay? She has Tay-Sachs disease or syndrome. What's going to happen here is that one of the enzymes that is going to be inside of the lysosome is going to be beta-hexosaminidase. And this hexosaminidase beta is going to be responsible to break down gangliosides. It's a type of lipids. This enzyme has a mutation, cannot do that. So these gangliosides are going to start to build up inside of the cell. They're going to start to build inside of the cell. They're going to damage all part of the body, but your nervous system is going to be the most vulnerable one and is going to show the symptoms first. And that's what's going to happen. This child will die because of that single lysosome enzyme is not able to do what it's supposed to do. So every one of those enzymes in the body is going to, in the lysosome, has a specific function to break down specific molecules. In this case, that is going to be autosomal recessive trait. Both parents gave the defective enzymes. That's why this type of diseases are going to be much more common among people who marry among their family members, or people who are, for example, they only marry among people from their own type or their ethnicity. You know, Ashkenazi Jews, about 4% of this birth that are being, among those people that intermarry Ashkenazi Jews, they're going to have this Tay-Sachs syndrome because they are going to have the defective genes more prevalent in their population. So if it is more prevalent in their population, if you're going to marry, there's a higher chance of having the defective gene passed on to the offsprings. That's why you see these kind of diseases that you usually don't see in normal population. You don't see it as many. But in certain population, you're going to have a lot of these kind of diseases. And so diversity is good. Okay? If some of us have been told that, oh, you got to marry your own kind, okay? You got to marry where, if you are from this country, you got to marry only these people from this country. That's a wrong idea. At least biologically, that's wrong. Diversity is a good thing. Diversity is a good thing. I'm very serious about that. You have no idea how prejudiced people are. You have no idea how prejudiced people are. I am coming from a culture that, you wouldn't marry somebody from another country. This is not something, that you would do. And that's a backward mentality. When it comes to diversity, diversity is good. The more diverse we are, the better we are. Don't listen to Donald Trump. He doesn't understand damn thing. Diversity is a good thing. Keep that in your head. Diversity is a good thing. Genetically is a good thing. Biologically is a good thing. Okay? Now, I don't know. If you marry somebody who is not, familiar with your culture, probably the rate of divorce is higher. I understand that. But at biological, foundation, diversity is a very good thing. Okay? Why do you think that purebred dogs have so many diseases? Why do you think that the beagles start dying at the age of 10 while normal dogs should go with 15, 16 years old? Because they are not diverse. Because they're all purebred with the same over and over, so that all these very rare genetic diseases are going to build up and build up and build up and ultimately causes disease that you don't see in normal population. I'm not saying that. This is a fact. This is known. If you talk to any geneticist, is going to tell you that. Mitochondria, powerhouse of the cell. But again, it does a lot of things. Pay attention. Mitochondria has two membranes. One membrane is from the original protobacteria, that bacteria that I told you long time ago was taken up by a larger cell. The second membrane is going to be what was left from that vesicle. So when that big cell ate up that bacteria, put it inside of the vesicle. They have remained. They are still there. A lot of things are going to be done by mitochondria, a lot of different things. But the primary thing that we need to, what I'm emphasizing here, is the production of energy, ATP. ATP is going to be produced in mitochondria. It's different than the production of ATP in prokaryotes, because prokaryotes only can make ATP by glycolysis. One glucose molecule, 2 ATP. Mitochondria can use the same glucose molecule and make 36 ATP. So definitely it's going to be a great advantage. Definitely is a great advantage to have mitochondria in your cells. You're going to produce more cells. That's why the eukaryotes are bigger in size. Because if you're going to be bigger in size, you're going to be able to produce more energy to support the metabolism inside that cell. That's why mitochondria is going to go ahead and produce a lot of ATP and allow the cells to be able to become larger and more specialized. It has its own DNA. Mitochondrial DNA has about 56,000 base pairs, nucleotides, ATGCs, okay? It has about 11 genes. It also has a lot of RNA coding, makes RNA molecules inside there, okay? And I don't want to get into details of it, but we know a lot about how these genes interact with the genes in the nucleus, okay? Some of the genes in the mitochondria were transferred to nucleus. You don't see them in here, even though you need to use the product inside of the mitochondria. Many of the genes that are going to be produced inside of the mitochondria, you use them inside of the mitochondria, not outside. But we know that there is a DNA, and we know that that DNA has a specific function, especially in the production of energy. So what happens if something goes wrong with the production of energy? What happens if you have a genetic mutation in one of those mitochondrial genes? Not the DNA in the nucleus, but DNA in the mitochondria. Lee's disease. Lee's disease is the time that you are having a mitochondria. One of those genes that I told you in the mitochondria has a genetic mutation in it. So what's going to happen is that there are two possibilities. You can have the Lee's disease because of one of those mitochondrial mutations, or you can have the Lee's disease when you are having the pyruvate kinase, which is going to be inside the mitochondria as well. What is pyruvate kinase? Pyruvate kinase here. Both are going to result in the same thing. Mitochondria cannot produce ATP. If you cannot produce ATP efficiently, where in your body you are going to see the harm first before anywhere else? Where is going to be the most vulnerable area of your body? Your nervous system. That's why. You see that child cannot move a muscle. That child cannot even suckle. That child will die within 2 or 3 years after birth. And this starts showing up, 3 months after birth. You're going to see all the symptoms coming up. Cannot move, cannot do anything. And this is going to become so bad, the brain is not going to get enough nutrients, and all those parts that are responsible for the movement, they're going to start to die because of the mutation in one mitochondrial gene. So you cannot undermine or underestimate the power of these genetic mutations. Now, all these genetic mutations are not going to be bad. Sometimes you are going to have good results too. But that's beyond this point. I'm just telling you how cell problems are going to be translated into disease. Anytime that you have a disease, there is a reason behind it. Most of the time we know what the reason is. Sometimes we don't. Okay? If we don't know it, that doesn't mean there's no reason. We just don't know it. Cytoskeleton. The cytoskeletal filaments, as the name indicates, they are going to be basically a skeleton of the cell. There is no bone there. They give the shape of the cell. They give the cell ability to move. They allow things to move around. When we are talking about cytoskeleton, again, if you put things in perspective, it's going to make more sense. Let me give you one example. I have a neuron. I have many neurons. One of these neurons, the cell body is going to be in my back. The end of the cell is going to be down to my toes. Now you got to ask yourself, that mitochondria has to travel from the cell body all the way to, how does it do it? How does it travel more than a meter in length? How does it travel 3 feet? It wouldn't be able to do it without the cytoskeleton, okay? There are different types of cytoskeleton. The 3 main classes are going to be microfilaments, intermediate filaments, and microtubules. The one that's going to be used to allow the transportation of mitochondria, lysosomes, things like that, is going to be microtubules. We're going to talk about these a little bit in these upcoming slides. But let me go ahead and give you a general view of it because they are very similar to each other. There are differences. I'm not saying they're exactly the same. There are differences. But they are very dynamic structures. They are very dynamic structures. Let me explain to you. If this is the cell body, I don't know how well you're going to be able to see this, but let me do it like this. If this is the cell body and this is an axon, a mitochondria needs to travel from here to here. What's going to happen is that it doesn't have a leg, cannot go ahead and travel on its own. It needs a path. It needs a road. What that's road going to be called is going to be microtubule like this. The way that is a fancy way. As the mitochondria is moving forward, the road is going to be laid in front of it. Okay? As passes, the road is going to be picked up behind it. So it's going to have a road that's going to enable it to go from A to B, but at the end, when it gets to the destination, everything is cleaned up afterwards. Because of this, these microtubules, proteins. These proteins are able to polymerize, which means come together or depolymerize, move away. If you have ATP energy, you would be able to energize them, and you would be able to assemble them and polymerize them. If you are done with it, all you have to do, not to add the energy, and they're going to be broken down and depolymerize. Okay? All these filaments, cytoskeleton, we're going to work basically the same way. There are different proteins involved, but the principle of polymerization and depolymerization is the same thing. Let me do something here quickly. Let me see whether or not I can find you folks. I should have done this sooner, but let me see if I can find it. All right. Let's see.
This time-lapse video recording is about 85 times. Also, the cell marked by the white arrow as it moves to the right.
See how actively they are moving? You see how these cells are moving? It's happening right now in our body. We just don't realize it. Cells are moving around, exploring their environment.
Notice how the ruffling movement changes as these two cells interact with each other.
All right. So when we are talking about these cytoskeletal proteins, all of them are going to originate from what we call these mitotic, in the cell we're going to call them centrosomes. That's you know, microtubule organizing center. MTOC is the fancy name for it. But we call it in the cells centrosomes. Centrosomes are going to be similar proteins. So if you are looking at it, you can see there's a central in here, and there's a central that's going to be perpendicular to it. Together we call them centrosome. Each one of them single is going to be centriole. Together we call them centrosomes. Each cell has only one pair, unless they're dividing. If the cell is dividing, you're going to duplicate your centrosome. One is going to go to one side, the other one is going to go to the other side, because when you're going to duplicate your DNA material and pull half of it this way and half of it that way, these guys are needed. When you're looking at here, the red is going to be centrioles, the centrosome. The green is going to be the microtubules. You don't see the DNA, but the DNA is going to be in the center. And it is going to be the job of the microtubules that are attached to the centrosomes to get half of the genetic material to this side and the other half to that side. So the cell is going to be divided, and now you got two cells. Okay? That's what it is. Very simple and straightforward. Majority of the microtubules are going to originate from centrosome. So if you're going to go and follow it out, where is it coming from? Where the story started, you're going to end up in there. But there is more to these microtubules and things like that. But I just want you to know that you only got one pair of centrosomes, and that pair is going to be responsible for originating the microtubules. Microtubules are going to originate from the centrosomes. Okay? When we are talking about these cytoskeletal, we have different examples. So when we are talking about cilia, or we are talking about flagella, those are because of cytoskeletal proteins. Okay. When we are talking about, imagine that if this room was a cell, in reality, what you have these intermediate filaments, which are another type of cytoskeleton. But intermediate filaments are going to go from the floor to the ceiling, from this wall to that wall, from here to here, because of the fact that they maintain the shape of the cell. You can ask yourself, how is the shape is going to be maintained? Because I just told you that the shape of the cell, the membrane is very moving, fluid. How is the cell able to maintain its shape? Why a neuron looks like a neuron, why a neutrophil is going to look like a neutrophil, is because of these cytoskeletal protein, specifically is because of the intermediate filaments. One of these intermediate filaments is the one that is going to be important part of what we call desmosomes. Does anybody remember desmosomes? Have you heard of desmosomes? Ask yourself. Let me give you an example. You know what bladder is, right? You know the urine goes inside of the bladder. But have you ever thought, why doesn't urine leak out of the bladder into my belly? It's a reasonable question. Okay? If I put water in my hand, it's going to leak out. How is this possible that the cells connected together, but is it unable to go through between the two cells? The question is no. Because you got these cytoskeletal proteins that act like a chain, like a stitch in between two cells. So now water is not going to be able to go through here because these cells are connected to each other like this stitching. Those are cytoskeletal proteins. What happens in here in the case ichthyosis, this one is specific ichthyosis vulgaris, is a disease that you have the mutation in one of those cytoskeletal proteins. These kids is a very rare genetic disease. It's quite common in England. I mean, all these three patients are going to be British. What happens is that epidermis of your skin is bound to the dermis by what we call hemidesmosomes. Same as desmosomes, just half the size. There's a mutation in those. What happens is that every day these kids wake up, they have to scrub their body and they lose their entire epidermis. So you see the skin because that skin is rough. And they go over this every day of their life until usually they develop some kind of infection and they die. But that's what happens because of a single mutation that is going to affect the function of these hemidesmosomes. They lose their entire epidermis every day. Okay? So this is the way that the disease is going to come. When you're having something fundamentally changed that it should not change. Cell division. So I hope that you guys remember it from the previous classes that you have had. Cells, we have two different types of divisions. One cell is going to divide and make two cells. That's going to be mitosis. The characteristic of mitosis is very simple. You are going to produce two daughter cells. They're going to be exactly looking like the mother cell. So if you are going to, how many chromosomes did we have? How many chromosomes did we have? 46. So if this cell is going to go through mitosis, you are going to produce how many cells? 2. How many chromosomes in each? 46. In the case of the other type, meiosis, or meiosis, some people call it, you start with the same thing. 46 chromosomes. How many daughter cells are you going to produce? 4. And how many chromosomes each one is going to have? 23. Half the genetic amount. And it makes sense because this is going to be an egg. You're going to have the same thing happening with the sperm. When the sperm and egg fertilize each other, you're going to have 23 plus 23, 46 chromosomes, and everything continues just like that. So these are the two main ways that you are going to divide the cell. Now, everything that is born needs to die. That's the law of nature as we know it today. Now, maybe 100 years down the road, you're able to download yourself in a computer and you don't die ever. But the bottom line is that right now you will die. Every cell in your body will die. Every living thing will die. But there are different types of death. Okay? There are different types of death. You got a programmed cell death. We call it apoptosis, Some people like to call it epitosis. Doesn't matter to me. Apoptosis is fine. And that's programmed cell death. It means that the cell death has been programmed inside your DNA. When you are you're going to embryogenesis. When you were just an embryo in your mother's uterus, what was that is that when you develop your arms, for example, when you're having your arm buds coming out, they're not going to look like fingers. This is the way that your arms are going to look like. They're going to look like paddle. What's going to happen here is that as you go through the embryogenesis, the cells in the center, they're going to go through apoptosis, programmed cell death, and they're going to die. And you're going to carve out your fingers out of the cells that are dying. If they don't happen, I don't know how long you're going to be able to see, because those cells in here didn't go through apoptosis because there was something wrong with something happened. This is what happens. Okay? That's a good way of cell death because of the fact that it does not initiate inflammatory response. Okay? It doesn't cause inflammation. What happens is that it makes the cell die, but it also going to change the characteristic of those markers on the surface of the cell. So when the macrophages are coming, they're going to know, oh, this cell is dead, I'm going to eat it up. It's going to eat it up, and they're going to take care of all the business inside of the macrophage. None of those lysosomes are going to be released into the environment, because the fact that when the cell is damaged and the lysosomes are released into the environment, those lysosomes are like acid that is released into the tissue. They're going to cause inflammation, and they're going to cause a lot of problems. That's necrotic cell death. So the difference between apoptotic and necrotic cell death is many, but in general, apoptosis does not cause inflammation. Necrosis, on the other hand, causes severe inflammation. If you go to, you don't see the picture. If you go stay in the sun for too long, you are going to go and get sunburned. You're going to lose your skin. All those skin cells go through necrotic cell death. And that's why you're burning and it's hurting and you cannot, lay down on your back, because of the fact that necrotic cell death causes inflammation. And that inflammation is primarily by releasing the lysosomal content into the cell, into the outside environment of the cell, interstitial space, and causing the cell death, inflammation, and everything that goes with it. So if you are going to go ahead and have a stroke and part of your brain is going to die, it's not going to die by apoptosis. It's going to die by necrosis. But again, you got another type of cell death, which is more programmed and controlled, doesn't cause inflammation. That's going to be apoptosis. All right. My God, we finished early. I was not planning to finish this early. So what should we do? Is there any questions? Can we answer any questions in here?
Just a question for the class.
Okay.
Do you focus more on case studies, or do you want us to memorize things or like systems?
There is always going to be a little bit memorization, Vinny, but I want you to focus on your comprehension of understanding. Usually I will give you a lot of case studies. Not everything is not going to be case studies, but I will give you case studies.
Will you give them for us to study from? Because I know in the book there might be several, but is there any extras we can get from you?
Oh, case studies is a matter of, it's like having if I teach you to drive a car, and I tell you the map to follow and be able to go and get, for example, to LACMA, the Museum of Arts in Los Angeles. You are telling me that, am I going to go and drive with you all the way to LACMA? I will not do that. I will give you the tool, I will teach you how to drive, and I will give you the map. You will have to find your own way. So you will see, it's not as hard as you think. Unfortunately, the way that I have noticed our education system is emphasizing on memorization. They don't care whether or not you can use what you're learning and apply. This is clinical aptitude sciences. This is a professional degree which really learning the information, everything that I have told you, I would have expected everyone in this class know everything I told you about today, so far. This I'm just repeating because if you don't remember it. I would expect a lot of things that you already know. The reason I'm repeating is not because I want to be redundant, because many of you have had these material a long time ago, you don't remember. So I want to refresh things. But whenever you learn the tools, when you know how to interpret the results, the rest of it is not as hard. If I give you a case study, I will not give you the case study twice. It's going to be a case study, and I'm going to make it up myself. So it's not something that I'm going to give it to you so you can memorize and learn it, because it's going to be different. But you will have all the tools. You will have all the tools to be able to interpret them. I promise you. You will see that. Because if you don't get a case study, you come to my office, we sit down and go over it, and say, did I tell you this, did I tell you this, did I tell you this? Were you able to put it together? I would assume that your first exam is going to be a little bit challenging for you. If you are going to be challenged, don't be surprised. Instead of fighting me, understand what I'm trying to go in and do, and understand, instead of trying to swim against the river, go with the flow. Understand how I'm trying to go in and put the emphasis. I don't put emphasis on memorization at all. I'm not a fan of memorization. That doesn't. I had this same problem. Somebody came, previous semester and told me, Dr. Nasser, I didn't learn this because he told me not to memorize anything. I'm not telling you not to memorize anything. I know, I need you to know what hematocrit normal range is, so you would be able to identify what anemia is. But I am not going to ask question on it, is this normal or is it like, those are kids stuff. You guys are professionals, so I'm going to go ahead and challenge you. That's a good thing, Vinny. That's not a bad thing. Okay? Any other questions? Okay, guys. I see those of you going to have lab in the laboratory, and the rest of you, have a good day.